So we were discussing the. Let me see. Let me share with you my screen. So we were discussing the article, tumor targeted delivery for. You have any questions or comments from our previous thing?
Mm, nothing from the previous one though.
You are sure with what we did discuss so far?
Yeah, I'm feeling pretty good about everything.
Wonderful. So now you know what is siRNA.
Hmm.
How does it work?
Yeah.
So just briefly explain me what how does it work?
So the siRNA is um basically
I'll call you in a while. Uh-huh. Okay. So yeah, so five. Yeah. Yes, yes, yes. Sorry.
let me just pull up the my notes from the last one. But um oh, where is that? Sorry, give me 1 second.
Yes, sure.
Okay.
Okay. So we are here.
Okay. Um, okay, got it.
Uh-huh.
So the siRNA is um basically the synthetically made double-stranded RNA um, that targets the gene to be silenced.
The target what's?
Sorry, what?
The targets what?
It targets the gene to be silenced,
Which part of the gene?
the non-coding region.
Targets messenger RNA.
Oh, the mRNA.
Yeah, it targets the
Yeah,
messenger.
yeah, I see that right now.
So which complex is involved in silencing that no messenger RNA? What is that complex called?
Which one?
So the siRNA destroys the messenger RNA by some enzymes, right? Some
Mhm.
proteins. What are those proteins? They are collectively called something.
The um.
Risk.
Oh yeah, the risk complex. Yeah.
Okay. So what is risk stand for? Like what does risk stand for?
Uh, I'm not sure what it stands for, but I know.
RNA induced silencing complex, right?
Okay.
So that is the reason I asked you that you should be familiar with what we have already studied because that is very much important in our next moving forward. If we don't know what we have studied so far, we cannot move forward,
Mhm.
right? So yes, and the things skip, but if you practice it like 2 3 times, so these are very easy things. Okay, so this was the topic. I mean, I asked you to read until this point before materials and methods. Did you read that?
Um, which, let me pull it up really quickly. Just need to be familiar. Um, up, up to up to the materials and methods.
Did you read that?
Yeah, I had to skim through it a little bit,
Okay, so you didn't read it like
not super thoroughly, but
yeah,
I
you didn't read it very carefully, right?
mhm,
So I will ask you to read it,
mhm,
and because we cannot move forward unless we read it, right?
mhm,
So you can do it right now. We can do it right now with each other, right? So
mhm,
instead of wasting time, rather than we can do it. So and I have to just I want to be interactive with me. Talk to me. If you have
mhm,
any questions, just talk to me and see what is that. Okay, let's see. The abstract is tumor targeted carriers provide efficient delivery of chemotherapeutic agents to tumor tissue. You understand it, right? CGKRK is one of the well-known targeting peptide with significant specificity for angiogenic blood vessels and tumor cells. So here is a new term which is angiogenic blood vessel. What is angiogenic blood vessels?
Mm, I'm not sure. I
So
haven't heard
what
of
is angio means? Have you heard of the word angio?
that. Yeah, I've heard of it with angiotensin one and angiotensin.
No, there is a common procedure, angiography, angiography. Have you heard that word, angiography?
Mm-mm.
No? When someone goes to the hospital with heart attack, they do angiography.
Oh, okay.
You know it?
I start to recognize it, but I don't know it.
Okay, so angio means blood vessel.
Mhm.
Angio means
Blood vessel.
blood vessel. So angiogenic genesis means
Um, the beginning of the blood vessel.
making new blood vessels. So we have to understand that when a tumor is formed, right? When a tumor is formed somewhere, so it's interesting thing to understand. Uh, Let me show you some tumor images, maybe tumor angiogenesis. Life is easy. You see this?
Mm-hmm.
So first, like there was a one tumor cell,
Mm-hmm.
okay, and that tumor cell got the energy from the neighboring blood vessel, right? And
Mm-hmm.
then another tumor cell formed, like it divided to make two. So now, how can this thing or this cell get can get the nutritions? So there, if there is like two, three cells, they can get the blood supply from the neighboring cells, right?
Mm-hmm.
But if there are more of the blood cells, if there are more of the cells, like this is a cell, 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 right?
Mm-hmm.
So how will they get the energy,
Um,
or the like? How will they excrete their waste substances, and how will they get the nutrition like oxygen and the food?
from neighboring cells.
Neighboring cells, but like the diffusion is not a viable option for the cells, right? So they need something. So they start making blood vessels
Mm-hmm.
inside the tumor. The tumor cells themselves start making blood vessels, right? And then those blood vessels, that is called angiogenesis. That is called angiogenesis. And then those blood vessels merge with the bigger vessel,
Mm-hmm.
right? And at some point in time, some of the cells dislodge from this place and enter into the blood, and from the blood they go to the other places. They go to the other places. So once they go to the other places, that is called metastasis, right? So we have to understand the term between tumor and cancer,
The,
cancer, right? So the tumor is something where the cells are there, but and then I mean it becomes like 100 cells, then 1,000 cells, then like 1 million cells. So when the cells mass increases, right? So they are not able to get the sufficient oxygen or food, or they are not able to excrete their food or oxygen. So what they do then, they start making blood vessels within them, right? And then those blood vessels start connecting with the previously made previously present blood vessels, right?
mm-hmm.
And then in during that process, some of the cells move into the blood vessel and go to the distant places. And when they go to the distant places, they they launch themselves there, and they start making again. That is called metastasis, right? And if there is more of the metastasis, then that is called, like, if there is like it spreads all over the body, then we call stage 4 cancer. But if it is like little, like two organs are affected or like three organs are affected, then it is manageable by surgery or by radiotherapy. But when it has spread all over the body, then it is not manageable. Then we have to do the chemotherapy, right?
Mm-hmm.
So you see that pro-angiogenic growth factors. So these are the pro-angiogenic growth factors, right?
Mm-hmm.
And then So you have to understand this. So when I read this, so he says CGKRK is a peptide, which is tumor targeting peptide, and it has significant specificity. It just binds with the angiogenic blood vessels. So it can bind with the angiogenic blood vessels. Here we designed fatty acyl conjugated CGKRK peptides. Now you understand what is the meaning of fatty acyl conjugated CGKRK peptide, or you don't understand? If you don't understand, tell me frankly. No worries.
I'm not sure.
Okay, not sure. You know what is CGKRK peptide?
Yeah, it's the cysteine,
Yeah,
guanine.
so it's five. Okay, so when we say fatty acyl, it means that there is a fatty acid attached with CGKRK,
Hmm.
right? So if you see that, so this is a CGKRK,
Hmm.
and then we have this chain, right? This is fatty acyl group.
Okay.
Okay, and now what is fatty acid?
Um,
Okay,
a long carbon chain, hydrocarbon chain with an oxygen attached.
now, so what is carboxyl fatty? Whenever the word fatty comes, fatty means something big.
Hmm.
Now we say that this person is fatty, means he's a big person, right? Fatty.
Hmm.
Acid means carboxylic acid.
Hmm.
So the long chain carboxylic acid is called fatty acid. So the long chain carboxylic acid is called fatty acid, right? The long chain carboxylic acid is called fatty acid. So we have, like, in this case we have 12 carbons, in this case we have 14 carbons, in this case we have 18 carbons, in this case we have 16 carbons, in this case we have 20 carbons, in this case we have 18 carbons, but we have an unsaturated bond at carbon number 9, that becomes oleic acid. Okay, so that is. So now we designed fatty acyl conjugated CGKRK peptide. Okay, based on the hypothesis that hydrophobically modified CGKRK peptide. So hydrophobically modified means, so this fatty acid is hydrophobic, right? Fatty acid is
Hydrophobic.
hydrophobic because it's like carbohydrates only, right? It's like it's a hydrocarbons. Hydrocarbons are all mm, uh, hydrophobic. They cannot dissolve in water, right? So hydrophobically modified CGK could enhance cell permeation. So they say that, so we can, if we have this peptide, it can enhance the cellular permeation and delivery of Si Rna targeted to the tumor cells for effectively silencing selected proteins. Got it? So what
Hmm.
did you get from here? We designed fatty acyl conjugated CGKRK peptide based on the hypothesis that hydrophobically modified CGKRK peptide could enhance cellular permeation and delivery of siRNA targeted to tumor cells for effective silencing of selected proteins.
So that means that it can, um, that syn- the synthesized peptide that they created
Mhm.
can, um, can go through the cells without really being inhibited by those extracellular factors that we had talked about.
Yeah, so it can enter. So this is synthesized fatty acyl six fatty acyl peptide conjugates using diverse chain of saturated and unsaturated fatty acids to study the efficiency of this approach. Add peptide to siRNA weight by weight ratio of 10 ratio 1, where N to P ratio is 13.6. I have explained what is N to P ratio. So the nitrogen represents the peptide, while phosphate represents the uh, what does phosphate represent?
The I wrote it down. Let me check. Mm, I can't remember.
Phosphate represents the we I explained you the phosphate represents the siRNA, because nucleotide siRNA is made from the nucleotide, and nucleotide has ribose sugar, nitrogenous base, as well as phosphate group,
Okay.
right?
Mm.
So almost, I mean, don't worry. I mean, I think you are overwhelmed from the previous class and you are just jumped into this class, so you are not able to connect with what we have discussed earlier, right? Do not worry, do not panic, okay? So just relax. Because in this science is all about concepts. So we have to build on one concept to other concept, right? And everything interplays here. What we have studied so far, that interplays here. That's why it becomes really hard for some people to study the research article, because they they cannot, like, they don't have that skill where they can connect all what they have read to the line they are reading. Okay, so now, this was important. N2P ratio is 13.6. Almost all the peptides showed complete binding with siRNA. How can I determine now? This is a very big statement. Showed complete binding with siRNA. What does it mean? How can I check that siRNA is binding with protein, with the peptide?
Mm. Well, if they were able to get the weight to weight ratio
No,
of
at that weight to weight ratio, like they took, like, say, 10 microgram of the peptide and 1 microgram of the siRNA, right?
mm,
Then all the peptides showed complete binding with siRNA.
mm.
How did they determine that?
maybe
How-
if
Yeah, yeah, maybe, yeah.
maybe if the weight to weight ratio increased, then they would be
Oh,
able
no,
to.
no. My question is simple. See, I say this hand is binding with this hand.
Mhm,
Okay.
mhm,
So it's completely binding.
mhm.
So you can see that it is completely binding. But if it is like this, I can say it's not completely bound.
Oh yeah. Mhm.
So my question is, they are claiming that all peptides showed complete binding with siRNA. How did they know the word complete binding? How do they know that this is a complete binding?
Um, if they were able to get the delivery of it into the cell, would that have?
So we have the simple answer is they must have designed an experiment,
Mm,
right?
mm.
So they must have designed an experiment. That experiment would have told them that the siRNA is binding completely with the peptide,
Mm.
right? So that assay is called, that experiment is called gel retardation assay or cyber green assay. We will discuss it later. Okay, and so they determined that, at which ratio they bind, at N2P ratio 10 ratio 1. I mean, 13.6, or weight by weight ratio of 10 ratio 1. So it means that, what this 10 reflects?
There's 10 nitrogen. The oh the
We are talking about weight.
peptides.
10 is representing,
The peptides.
and 1 is representing what?
The siRNA.
Gas, right?
Mm,
So it means 13.6 times more nitrogen than the phosphate,
mm.
right? And then this was the first experiment they did. And at weight by weight ratio of 20 ratio 1 and by P 27.3, complete protection of siRNA from early enzymatic degradation was observed. How did they observe?
Another experiment that they did
Another
then.
experiment, yes. So we have to learn another experiment.
Mhm.
Then conjugated peptides and peptide siRNA complexes did not show, this is the third line they are saying, did not show significant cytotoxicity in selected cell lines. Now, how did they do? How could they know that they are not showing significant cytotoxicity?
Another experiment.
Experiment, right? So yeah, there are three experiments in three lines. Then, then they say The oleic acid conjugated peptide showed the highest F, but we should know that, I mean, what information we are getting from this experiment, that siRNA is, first time we check that either siRNA is binding with the peptide that we have made. Second, they wanted to check whether that peptide can protect the siRNA, right, in the blood. And third, they checked whether this siRNA peptide complex is safe for the cells or that is not safe to give to the patient, right? So these are the three experiments. Then oleic acid conjugated peptide showed the highest efficacy of siRNA uptake. This. Now this is another experiment. How did they know that oleic acid conjugated peptide showed highest efficiency in siRNA uptake? So siRNA uptake is another experiment. This is the fourth experiment. Uptake, right? So what do you mean by uptake? Uptake means whether this peptide siRNA complexes are entering into the cells, right? If they are entering into the cells, then we have to determine how much they are entering. How will we know that? Again, an experiment. And then they said that they silenced kinesin spindle protein at peptide siRNA ratio of 80 ratio 1, 109. This is another experiment. How can I know that kinesin spindle protein has been silenced? This is another experiment. The siRNA internalization into non-tumorigenic kidney cells were negligible. That is very interesting. They say that the siRNA was not able to enter into normal cells, non-tumorigenic kidney cells. They were not entering into the normal cells, but they were entering into the cancer cells. So what does it mean?
That means that they were able to find that they can target the cells
They
that
can target the cancer cells. So when I administer that complex into the body, it will specifically go to the cancer cell, but it will avoid the normal cells.
Mhm.
That is why siRNA internalization into non-tumorigenic kidney cells was negligible. It means the siRNA peptide complex, because the normal kidney cells do not have the CGK-RK receptors that can help internalize the complex into the cell. Then these results indicate that conjugation of the fatty acid to CGK-RK could create an efficient delivery system for siRNA silencing, specifically in tumor cells. That's it. So what is, if you, so this is basic science. So basically they hypothesized, first their challenge, what was the challenge? To deliver siRNA into the cell. Okay, this was the challenge. Then they designed what? Peptide. And they conjugated that peptide with fatty acid. Okay, so peptide fatty acid conjugate. Now that peptide fatty acid conjugate is, we want to know whether this peptide binds to the siRNA or no. We have to design an experiment to know that whether this peptide binds with the siRNA or not, right? One experiment. Second, they wanted to know whether this siRNA peptide complex will survive the blood. It will survive. I mean, if I give siRNA, so will it be destroyed by this, by the blood and nucleases or different enzymes that we talked about in the last session, the barriers to the siRNA delivery, right? So that was the second, which is called serum stability. So in other words, complete protection of siRNA from early enzymatic degradation was observed. This is called serum stability. First is binding, second is serum stability, third is cytotoxicity, fourth is uptake, and fifth is silencing. So how many experiments did they do in this paper?
5 total.
Five, five. And what was the sixth experiment? Making the peptide and siRNA.
Mm.
Making the peptide with hydrophobically modified, like fatty acid. So overall, six experiments were there, right? So if you do those six experiments, you are going to be the next scientist, right? So now I stop it here for good, and I ask you to please read this from here
Mhm.
to here.
Okay.
Got it?
Mhm.
And once you are finished with reading, and once you are finished with what we have studied so far, okay,
Mhm.
then we will move forward to the next step.
Okay.
Does it make sense?
Yes.
Or no?
Yeah, it makes sense.
And I will like you to please explain me. So next time I will come, I will be silent, your student, I will become your student, and now you will become my teacher, and you will explain me. Imran, see, it's not difficult.
Mhm.
We have a person who got Nobel Prize by discovering siRNA, right?
Mhm.
And what is siRNA? I tell you. Then whatever the word you speak, I will question that word. If I say angiogenesis, I should know what is the meaning of angiogenesis. Here are different words you can see. Post transcription process of protein synthesis. Now it's a difficult word, but if you have understood the previous three sessions, this shouldn't be a difficult word. Then uh, nucleases in the serum. If you have understood that, you can easily understand that, right? Then, for instance, uh, risk, right? Have
Mhm.
we discussed that earlier?
Yeah, we did.
Okay. So what is siRNA? Double stranded siRNA is 23 nucleotide, and the antisense strand directs the RNA-induced silencing complex to target messenger RNA. What is this note 2? So yes, then you have to see what is mitochondria.
Yeah.
You know what is non-tumorigenic cells.
Mhm.
For example, arachidic acid. Okay, and if you read that summary of the biochemistry that I gave you in my first session, like
Mhm.
26 pages of biochemistry summary, right? Huh?
Mhm.
If you read there, you will easily understand fatty acids. Simple. Long chain carboxylic acid, saturated fatty acid, unsaturated fatty acid. You should be knowing. Like if I ask you oleic acid, you should be knowing. Oh, it's 18 carbon fatty acid, which has uh, unsaturation at carbon number 9. If I talk to you about omega 3 fatty acids, have you heard about omega 3?
I know that it's found in salmon
But what
and
is this omega 3? From where did it come, omega 3?
um, I have
Right.
no idea. I don't
So,
know.
see, this is scientist. So, basically, this is the difference between a scientist and a layman. Scientists, I mean, I think I told you earlier, scientists are paid for being children, right?
Mhm.
Because they ask questions. They don't take things for granted. They don't accept anything.
Mhm.
They they don't accept. I mean, they don't accept unless it is proved by an experiment. Okay,
Mhm.
that is basic science. They will not expect. They will not accept any statement made which cannot be proved by experiment. Got it?
Mhm.
And learning this, it will be. I mean, it will be fun. It will become fun for you if you really understand it. Like, what is this MDA MD 435? I mean, difficult terms. AU 565. What is that? What is HEK 293? What is Malditov? What is RPHPLC? If you don't know, you are free to ask me question. Just ask me question. I don't know. Don't take it for granted. I mean, you have to grab me from my shoulder. I mean, you didn't tell me. You have to tell me. Got it?
Mhm.
Does it make sense to you?
Yes.
What is your job now?
Now my job is to go over um, the rest of the introduction or abstract for this,
Mm-hmm.
and then go over the biochemistry pages that you sent me,
Mm-hmm.
um, and just review the process of
Mm-hmm.
um,
Yes, I tell you one thing, Sarah. I tell you, it's very fun thing. Take time, revise that. When I started, I have something in my mind that you cannot see right now because you cannot see. But once you spend time with me, you will see that. and you will start enjoying. Wow, you will start saying, "Wow, I should have known it earlier," right? So once that wow factors comes in you, then I will be out, and you will become an independent scientist if you want to become a scientist. Yeah, what do you think? Yeah.
Understand. Yeah.
Okay. So we finish this here. We will. I mean, I will be waiting for your email. With when you are done with this.
Mhm.
Okay. Previous three sessions, my notes, and this thing.
Mhm.
Okay. So then we will move forward in our life.
Okay.
Okay. And I will be waiting for your feedback, comments, suggestions, because if your feedback, your engagement will give me morale, like give me some boost to move forward, right? Because it
Mhm.
is you. It depends upon the receiver how much he wants to receive or she wants to receive, right? Got it?
Yes.
And don't get bogged down. Everyone, I mean, I was like you when you, I mean, I was your age, so don't get embarrassed. I was like I was similar. I mean, I was. It is normal. It's a very normal thing, right? So
Yeah.
don't feel like I don't know. I didn't mean that you don't know. You know everything. Just need to be a little aligned, connecting the dots. Once you connect the dots, you will start appreciating. Yes, sometimes we take little things for granted, but little things have more meaning than the big things.
Definitely.
All right. Alrighty. Any questions?
I just want to just clarify. Um, so after I finish like reading all of this and then going over just the previous stuff so I can connect everything together,
Mm-hmm.
then we can set up another meeting
Mm-hmm.
for later on.
Yeah, and then I will explain you the experiments.
Okay. Okay.
So I
Great.
will explain you the experiments, and then you will come to lab to do those experiments.
Okay.
All right? Are you up for that?
Yeah.
Down.
It sounds fun. So,
Sounds fun. Okay. Let's see. I mean, because I mean, this scary stuff, it will become piece of cake for you.
mhm,
It will become, these scary diagrams. These will become piece of cake for you if you
mhm,
understand that. I mean, this will become piece of cake for you. Believe in me. This will. I mean, so what? This is nothing. But if I expose you directly to this, you will say, sorry, this is very difficult. I cannot do science. Sorry, science is very difficult, right?
mhm,
Science is not difficult. The way we approach science is difficult. We should approach the science from the basics,
mhm,
right? We should see science is something that happens. Science is not something that is not comprehensible, right? It is not metaphysics, right?
mhm,
It is metaphysics means anything for which we don't have any explanation. There are billions of questions, so we human beings don't know answers to, right? And those questions are waiting for you people who, if you get trained in that way of doing things, then you are an next Nobel laureate. But it depends upon you.
mhm.
Okay. Anyways, uh, thank you so much for joining me, coming and showing up on time. I will put this, and this is this is an experiment that I am doing. These recordings are with you, and if you keep following that, because I have done that already with Sean and Kaylee, right? So I know what's the same thing. It's the same thing for me, right? But and should not get discouraged. Okay.
Mhm.
Next time, you have seen the sportsman.
What was that?
Sportsman. Did you see the sportsman? Like
Mm.
sportsman, like any sports, like
Oh,
volleyball,
yeah, like soccer. Yeah.
like soccer.
Mhm.
So is it possible for the athlete to perform to its best in every session?
No. I mean, yeah, but
No.
they,
Yeah,
you
no.
be
The
super
one like Kobe Bryant, I mean, name any of the top person in that field.
messy.
Messi, yeah, Messi. So do you think that Messi became Messi in one day?
No, absolutely not.
He must have made a lot of mistakes,
Yeah.
right?
Yeah.
And you and me, we are here to make these mistakes, to become Messi of science. Not Messi, that Messi. He's Messi person, not that Messi. Okay, have fun. Bye bye.
Bye.
Bye.